All right, today we're going to be looking at the Anatomy and Physiology 1 Histology objectives. Now, it's not going to be all of them, but we are going to look at a few. But some notes before we get started is that this video should not be your only resource for study. There's lots of great resources on campus, and you also have your lab manual, professors, and tutors at the BSc. Speaking of the BSc, the Biological Science Center is on campus, and it's a great place to study and ask for help. We have whiteboards and markers and slides and just everything you might need. And obviously we have tutors there that are so ready to help you guys out. And while you're there, please use proper lab etiquette and treat our lab microscopes, slides, models, etc. with care. You can also request oil for oil immersion to see things closer up, but that is upon request just to make sure we're not damaging our microscopes or our slides. And then one of the last notes is reach out to your professor if you have questions about this video, questions about your objectives, just any questions at all. We're here to help at the BSc, but also your professor is going to be one of your greatest resources. So to start us off, we are going to look over 4 different categories of tissue: epithelial, connective, muscle, and nervous. So to start us off, we're going to look at epithelial tissue. And something that I think people really struggle with epithelial tissue is knowing where to look. For a lot of these slides, you're going to be wanting to take in the whole picture, but for epithelial tissue, we're just looking from the apical to basal surfaces. And what I mean by that is that epithelial tissue typically lines a cavity or is on the surface. And so we're looking for where that kind of basement membrane is to where that cavity or surface is. And so we can see here that this right here is going to be that basal surface. It's kind of this dark line that goes all the way around. And then our apical surface is going to be facing this lumen right here. And so what we're looking at is actually going to be these cells right here. And they can go all the way around right there. And this is going to be our simple squamous epithelium. Looking here, we have simple cuboidal. And the reason that it's simple is because there's typically only one layer involved. And again, we're going to be looking from the basal to apical surfaces. And so here we're going to see this, that clear outline right here on this one. Those are going to be our basal surfaces, and our apical surfaces are going to face the lumen, which is going to be this right here. And so we can kind of see, especially in all of these, they're kind of circular. On some of these we have a bit more of an elongated shape, but here we're going to see we have one layer of cells facing the inside, have our basement membrane right there, and this is going to be simple cuboidal. You can kind of tell they look a bit circular sometimes. We call them cuboidal because sometimes they look like squares. Next up, we are going to use that same technique, right? We're looking at the apical surface, the outside, to the basal surface right here. You can tell there's a difference between this cell and these cells down here. But this is going to be our simple columnar, and you can tell because we have these long, thin nuclei, right? We also see a bit of cilia around here, and we can tell that this is one layer, right? We're not getting too messy yet. We just have a simple layer going on there. So this is simple columnar. This is going to be our keratinized stratified squamous epithelium. And so keratinized, the way I like to think about it is that these cells are dead, right? We can see that there's kind of like a hole where our organelles should be and things like that. And we can see it gets really thin, really flaky. These are often pretty squished and they're kind of just devoid of life. So there is that. And then for this is still going to be that stratified squamous epithelium, but this one is going to be non-keratinized or not dead. And so we can kind of tell the difference between these two. We can see these cells are a bit more full of life, and they kind of do still thin out as they get to the top, but we can tell they are not quite like this. This, we can see a lot more empty than this. This is a lot more full. But again, we're still looking from that basal surface up to the apical surface. And here's just another example of that. This is the cavity lining, so there's some on both sides, but we're just primarily looking at this stuff right here. Now this one is a bit of a tricky one. This is going to be our transitional epithelium. And the way we can tell that it's transitional is because of the fact that we are seeing a lot of variation in these cells. Whereas a lot of them on the other ones we've looked at prior, they're very uniform. Transitional, lots of variety. Lots of variety. And typically you can tell the cells change from one shape to another as they go from bottom to top. Here's another example of transitional, and also another example of why it's so important to know where you're looking at for epithelial tissue slides, because all of this is a different type of tissue than this right here. So it's always important to look at where the pointer is pointing at on your microscope, and also know and be able to recognize epithelial tissue. This is another epithelial tissue. This is going to be pseudostratified columnar, and this is going to be ciliated right here. This is our cilia all the way around here. We can tell it's a bit more pronounced. We did see cilia in our simple columnar, but here we can tell it's a lot more pronounced. And the reason this is called pseudostratified is because it looks like there's two layers, but there's really not. And so you can tell here, this looks like its own layer of cells, and this looks like its own layer of cells, but they're really just one layer. The way you can tell pretty easily is you can follow the cell wall here and try and make out where these cells are really coming from. This is just another example. There's lots of different ways that this can look, but you can tell it's pseudostratified because of its really pronounced cilia and then just following that cell wall. Getting into connective tissue, this is going to be our areolar tissue. It is really stringy. It kind of looks like a bit of a hot mess, but it is recognizable by its lots of fibers. Lots of fibers here. Next, this is one that I think a lot of people struggle with sometimes, but this is going to be reticular tissue. One of the main ways that we can tell it's reticular tissue is that we are going to have these really pronounced fibers running all the way through reticular tissue, and we're going to have lots of these little dots scattered all the way around reticular tissue. So we can see these fibers, these little dots. This is one of my favorite slides, but I'm going to show you a few different things about reticular tissue and why I really want to emphasize coming into the lab and looking at the slides yourself, because there's lots of variation within slides, and so if you're only ever looking at one example, that's not really going to do you good. It's not going to help your studying. So one thing people struggle with a lot is the difference between some reticular tissue and blood. So I want to point out that example really quick. So blood is another connective tissue you're going to have to know. We'll get to that more later. But right here you can tell that there's nothing really inside of these cells, right? Red blood cells kick out most all their organelles just to carry oxygen, right? And so we can see we have all of these, they're completely empty. Whereas on the reticular tissue side, first of all, we can see these fibers stringing through, right? We can still see those a little bit. And then these cells have a lot going on inside of them, right? They're not empty, they have a whole party going on. And where it might get confusing is you can see these things running around here like little platelets or different types of cells within the blood, but what we're really looking at is there something inside of it? If so, reticular tissue. If not, you might be looking at blood. And this is another big example of why I really emphasize coming into the lab, because this is also an example of reticular tissue. It's definitely different colors. Some people go off of color when they're studying, but that is not always your best friend, because the colors can really vary depending on how the slide was preserved and stained. So we really emphasize getting to know the structures rather than colors, because that can change on you. So here, we can definitely still see these fibers. We're definitely still surrounded by these little dots you see everywhere. But yeah, I just say, just definitely know those fibers, definitely know what these little cells look like. That is important for reticular tissue. Next, a lot of people struggle sometimes with this one, but this is going to be dense, regular connective tissue. And on this note, I really want to emphasize, ask your professor how they want you to write your answers. If your upcoming exam is a little confusing, ask beforehand if they want you to write it all out. So what I mean by that is writing just dense regular. That might not be enough. You might need to write dense regular connective tissue or something like that. And be sure to read the questions very carefully on your exam, because it could be asking you for general or specific tissue type. If it's asking you for general, this would be connective tissue. But if they're asking you for specific, just clarify with your professor if they want you to just write, dense regular or dense regular connective tissue, just to be as precise as you possibly can to make sure you're not missing out on any of those points. Next, we're going to have our dense irregular connective tissue. And you can really tell this one is a bit more sporadic than our other one. We can tell we have a whole lot going on here. This is a personal favorite of mine. I just think it looks really fun. But definitely note the difference between the two, and I'd take some time to look at both of these slides carefully. Next, coming up, we have adipose tissue. Now, adipose tissue is fat, and so you can see this vacuous space right here. Some people think this looks like bubbles. Some people think it looks like a spider web. However it sticks in your brain, just remember that this is adipose tissue. Coming up next, we have our hyaline cartilage, and if you'll notice, around the edges, we have that adipose tissue. That's another identifying tool sometimes, is being able to know what kind of tissue surrounds the thing you're looking at. But when we're looking at these cartilages, the main things, we're looking at this strip right here, right? We're not taking too much care to this around here. I just thought I'd point it out, but we're mostly just looking at this strip right here. And a tip for identifying hyaline cartilage is going to be that it's very glassy looking. It's very smooth. Very, very smooth. We have all of these little holes right here, but we're really just looking at the smoothness. And we're going to come up on a different slide here, our elastic cartilage. Some people get these kind of confused because they're similar in some ways, but I'm going to show you the difference. But again, we're just looking at this right here primarily to help us identify this tissue type, this kind of rectangle right here. You can tell there's still these holes, there's still this, texture right here, but let's look at this closer. Right? So the way we can tell these apart is that our elastic cartilage is going to have these fibers, right? They're really pronounced. They're really there. We can tell it's like these spider webs again, right? But on our hyaline cartilage, so smooth, very glassy looking. It is, no fibers. We can see some of them on the inside, and I think that confuses some people, but we're looking for the space between the holes. You can see that difference. Lots of fibers. No fibers at all, almost blurry. All right, next we have fibrocartilage, and this is one I also definitely recommend coming into the lab to look at because there's lots of different areas on this slide. This is just one section of a much larger slide. This is one of my favorite slides actually, but we can tell, right, we have these ripples going on. We see lots of stuff going on here. It can be really confusing to look at. So I recommend coming to the lab and looking at this one. Again, this is fibrocartilage. Next, we have bone. Everyone loves bone. It is just super easy to work with here. But what we're really looking at are going to be all of these osteons right here. And you'll get in more of this with your upcoming bone exam. You'll have to identify the parts of this. But right now, just focus on knowing that this is bone. Another helpful thing that people tell me and that I have started is that it looks like the trunk of a tree, right? So that's how we know that it's bone. We can see we got these rings that are kind of radiating from the center. So a lot of people think of this as a tree trunk, and that's a helpful way to help them remember that this is bone. Next, we have blood. Circling back to it from earlier, again, we can tell that there are no organelles in here. They're kind of just blank cells, right? There are red blood cells. But we can tell there's a lot of different stuff around here, but primarily we just got to know this is blood. You'll get into what these are in A&P too. Next, this is going to be our smooth muscle. Now, a lot of people mix up smooth muscle with dense regular, and so I really recommend looking at the two side by side, kind of like we've been comparing with some of our cartilages and reticular and blood. Kind of take some time in the lab to compare and contrast the two so you can be able to tell the difference on your exam. Next, we're going to have cardiac muscle. Now, this one can sometimes get confused with skeletal muscle, but we're going to go through a couple ways you can tell the difference. So first of all, we're going to notice that there's lots of nuclei in here, right? We see lots and lots of cells, but we also see amongst the striations, or these little lines right here, right? We can see these are all striated, lots of little lines, especially down here, the lines are quite pronounced. We also have these right here. Now, these are going to be called intercalated discs. Right here, and they appear to be really, really dark striations. Here's another example of it. This one I really like a whole lot because you can really tell that is an intercalated disc. There's so many of them. You try and point them all out, right? There's so many. There's just so many, and that's going to be the biggest clue to being able to tell if this is going to be cardiac muscle. Next, we're going to look at skeletal muscle, and this is a bit farther away. You might see it like this, or you might see it this close up. So we can tell that we have our really, really dark striations, and there are some areas of the slide that are lighter, there are some areas of the slide that are darker, but we don't see those really, really pronounced dark intercalated discs. They don't really exist in skeletal muscle, right? And we can also tell in these skeletal muscle slides, we have very, very few nuclei, whereas in cardiac muscle, we can tell here so many, and we have those intercalated discs. So when we're looking at this, we know it's skeletal muscle because of the fact we have many, many striations, few nuclei, and then going back to smooth muscle, there are no striations in it. All right, so one of the last slides that we have for the main objectives is going to be your nervous tissue. And basically what we're looking at here is we're going to be able to identify the parts of a neuron. And so you'll have to know things like your dendrites, your axon, neuroglia, just parts like that, your soma, your nucleus. And we can walk through some of those, but you'll have to know it a bit more, like bone, you have to know it a bit more later on in A&P1 when you get to your special senses unit. You'll have to identify these parts and a bit more parts as well. We can tell these are all of our dendrites, they're going out, this is our little axon going this way, all these little dots are our neuroglia, and kind of just take some time with these neurons and practice if you can identify all of these areas. Here is another example of a neuron. So we have a couple different slides in the BSc. Some of them are stained pink and purple and blue. And so looking at all of them and kind of practicing, can I identify the parts? Do I know what I'm looking at? And kind of testing yourself like that or asking a tutor for assistance can really help build that knowledge. And so next we're going to look at mitosis and the stages of mitosis. So let's get into that. All right, so in looking at mitosis, there's a couple important things to note. First is we're going to be looking at mitosis in both plant and animal, so don't just look at one or the other. Look at both and try and compare and contrast the way mitosis looks. What we're currently looking at right now is stages of plant cell mitosis, but we're going to get into animal cell mitosis here in a bit. It's also important to note the different stages of DNA because that's one of the terms I'm going to use as we talk about mitosis. Chromatin is DNA in its loose active form, and that's really going to be during interphase and kind of the last part of telophase. We are also going to see DNA condense into what people typically recognize, that classic X shape or the chromosome. That is basically during prophase and metaphase. And then we're going to have our chromatids or our sister chromatids. That is really going to be an anaphase and kind of the first bit of telophase. And that we'll go through that more later. And just to touch on the stages of mitosis in order, we're going to go from interphase when the cell is at work, to prophase when all of the chromosome is condensing, and then metaphase when all of our chromosomes are lined up along the center of the cell, to anaphase when those chromosomes are split apart into those sister chromatids or just chromatids, whichever one you prefer. And then telophase when we're starting to form back two individual cells or identical daughter cells. So let's go ahead and jump in and start practicing identifying the stages of mitosis. So what we're looking at right here, we have a few different stages, and it's always important that when you're looking into the microscope, trying to find the pointer in the microscope, because whether I am pointing to this cell here or this cell here, those two are in different stages of mitosis, and so it's very important to make sure you're looking at the right cell when trying to answer those questions. So all of these cells here that you don't see, these really dark lines, all of these are going to be in interphase. So basically we're having chromatin and the cell is currently at work, whereas in these cells we're starting to see really dark condensed chromosomes. And so that is going to be the difference we see here between interphase and prophase. Moving on to the next phase, we can see that all of those chromosomes are aligned down the center line of the cell. So this is going to be metaphase right here. And we can see other phases as well, but we're looking here at this metaphase. Next, we have anaphase. And anaphase is when those chromosomes get pulled apart and they have their own individual sister chromatids. And so that is when they're split. Now in this phase, or in this slide, we can see many different phases, and you can just practice trying to identify the ones we've already covered so far. And then our last phase is going to be telophase, and I included this picture because we can see that this is going to be our anaphase, and this is going to be our telophase. And you might be thinking, well, that's really hard to tell the difference, but the key difference between the two is this little line right there. That is going to be the cell wall, and that starts to form when we are saying these are two individual daughter cells now. So we're starting to close it off. These are two different cells. They're no longer one set of DNA. They have separated, and they are daughter cells, so they're the same genetic information. They're just two separate cells now, two different cells. Here is another great slide. Kind of just practice what all stages do you see? Why do you think they're in those stages? And kind of practice a little bit with this. Here's another one, just to get a good look at the different examples. All right, and now we are going to move into our animal cell mitosis. Now these look a little different, they're a little less uniform. But we can see quite a few stages here, and you might recognize a few of them right off the bat, but let's kind of walk through them. So I'm going to point out a couple that I see here, right? We're going to have our interphase, our cells at work, right? We have that chromatin going on right here. I can also start to see we're going to have prophase, right? When we're beginning to condense that chromatin into chromosomes. I can even see here, this is going to be some really late telophase. Right, and we'll get into a bit more telophase tips here in a minute, but that's just a really great example to point out right now, I think. Next, these are a few different examples of metaphase, and you can tell they're quite down that center, and I think metaphase is one of the easiest because they're in the middle. I think M for middle, right there. All right, here's a few different more examples of metaphase, just to kind of let you know that it's also important to look at different slides when you're looking at mitosis as well, because they can really look different and you never know what you're going to see on your exam. Here is a great example of anaphase. We can see those chromosomes have been pulled apart into those sister chromatids, and they have split, and they're going to start being their own cells here in the next stage. Here's another great example of anaphase right here. You can see some others as well. All right, and so here is where we have that same problem. Maybe we can see that this is going to be anaphase and this is going to be telophase, but you might be wondering why, we can't really see a cell plate or cell wall, but what we can see is what we have in animal cells, which is called a cleavage furrow, which is this little indentation right here to show that we're starting to separate into two different cells, right? And so here we can tell this is anaphase because it's just one big circle. But right here we've started to enter into telophase because we have this divot, right? And we can see it on both sides, this little divot, and it's going to kind of pinch off from each other and form two daughter cells. All right, that is going to be it for this video. Thank you guys so much for watching, and don't forget to come into the BSc and use your resources for studying for your next exam.